Institute for funding this work. So today I will be talking about work with human data, mostly brain imaging and genetics. And actually this is a really nice follow-up from Lauren's talk. So we'll be talking a little bit about chromatin as well. And my collaborators from the Kavli grant are listed below. So the quest to understand the human cortex has been a very important but challenging one, and I think it's what has brought all of us together at the Kavli. And it's hard to argue that the human brain isn't unique. We share a lot of DNA with other species, such as the mouse and chimpanzee, but clearly our brain has expanded in a very unique way to underlie our cognitive capacity that is unique from other species. But just looking at the DNA itself isn't enough. So we need to understand what is shaping our cortex at the level of the genome. So to look at this cortical expansion, we are going to be focusing on two cortical measures, namely surface area and cortical thickness. And we can get these measures from magnetic resonance imaging, which now we can get on thousands and thousands of individuals quite quickly and a little less expensive these days. So surface area here is denoting this area of the cortical ribbon in blue, and cortical thickness is this distance between the inner gray-white matter boundary to the outer peel surface in turquoise. So the aims of this project are threefold. The first is to use genetically informed atlases to increase discoverability of the genetic variants underlying these two measures of thickness and area. And I'll get into the genetically informed atlas in a moment. And we also wanted to see whether these genetic variants generalize to an adolescent sample, because typically a lot of these sorts of studies use older adults. The second aim was to look more at the biological interpretation of the genetic loci underlying the cortex, and also looking at enrichment for different functional genomic partitions with a focus on these regulatory regions. And the third aim was to do this deeper dive into the biological interpretation of these regulatory regions by using cell type specific accessible chromatin sites and how that overlaps with our uncovered genetic loci, as well as looking at pleiotropic variants. So I will describe all of those in a moment. So for the sample, we used 40,000 older adults from UK Biobank. So this is really a big data study, and we have genetic data as well as brain imaging data for all those folks. And we also used the adolescent brain cognitive development study, including about 9,100 9 to 10-year-olds from the United States. For the genome-wide association studies, for those that aren't familiar, basically we are mapping single nucleotide polymorphisms, or SNPs, to our brain phenotypes of interest. So we have these common genetic variants all along the genome, where at particular positions along the genome, different individuals might have slightly different variants. And we want to link that to the area and thickness of these 12 color-coded regions here, where we have a unique solution for area and thickness. So these are these genetically informed atlases. So you can think of any sort of map. You can trace boundaries based on politics, based on elevation. So similarly with the brain, we can parcellate based on things like genetics, which is what we do here. Whereas commonly in other studies, people have used markers such as gyral folding patterns. So we have this unique spin where we have these genetically informed atlases. So for the main GWAS results, the numbers that you see on these brain maps here are the number of hits or significant SNPs that we find associated with area and thickness of each of these regions. And what should jump out right away at you is that we find more significant loci underlying surface area than we do for thickness. And there's some regions that have many significant loci, such as the occipital area and sort of some of these temporal regions here. Most of these variants actually generalize to our neurodevelopmental sample. So these maps were generated based on our discovery sample from those 40,000 older adults, but we do find that a lot of them are still significant in our ABCD childhood cohort. And just to put some more biological meaning to these significant SNPs, basically we took the significant SNPs for each region and we put them into a gene ontology analysis so that we can understand how these SNPs contribute to different biological processes. And we actually were able to pull out quite a few significant neurodevelopmental processes, even though we weren't necessarily searching for neuro terms. So for instance, just focusing on occipital area, in red in this top plot here, we see enrichment for biological processes like tube development, pigment cell differentiation, growth factor signaling. For parietal regions, we see some enrichment for Wnt signaling pathways, which is really a conserved pathway that we see in many, many species. And we also see enrichment in prefrontal thickness regions for things like cortical tangential migration and programmed cell death. So moving on to more of this regulatory feature aspect, we decided to run a partitioned heritability analysis. So essentially heritability is measuring the variance or the variance in a trait that can be explained by genetics. But here, rather than just looking at the entire genome as sort of a single entity, you can partition or compartmentalize the genome into different functional annotations. So for instance, you could look at specifically conserved parts of the region of the genome versus those that might be more human specific. So we focused on partitions of the genome that have been shown to have differentially methylated regions. So Lauren talked a little bit about methylation and this chromatin structure, but basically these methyl groups along DNA can cause chromatin remodeling, and it can also affect downstream gene expression. And there was a paper by Gochman et al. In Science that actually mapped out these differentially methylated regions, comparing modern day humans to Neanderthals and Denisovans. So just as a schematic, if you take those differentially methylated regions differing between ancient and modern day humans, we actually find that those DMRs are enriched for perisylvian thickness, which is this light purple region here. And what's really interesting about that region is that it's really important for language. It encompasses Broca's area, important for speech production. So already we're kind of getting at this unique human genomic compartment being associated with a very unique human brain region and function. So going even deeper into these regulatory regions, well, what's regulation got to do with it? So we looked at open chromatin or accessible chromatin sites, and this was in collaboration with Dr. Kun Zhang at the Department of Bioengineering, as well as with an undergrad student at the time, Jenny Dong, and a grad student, Yan Wu. And essentially we can map these accessible chromatin sites with single cell RNA sequencing techniques. And again, these accessible chromatin sites would allow transcription factors to bind and will impact downstream gene expression. So we've kind of already done this GWAS bit, going from genotype to phenotype. And in the next slide, I'll show that other papers have shown how we can go from genotype to these accessible chromatin sites in a cell type specific manner. And our analysis was really trying to bridge this gap here to our brain imaging phenotypes. So in previous studies, we took the data from these studies looking at primary motor cortex tissue, where transcriptome profiling was done in this tissue to get these 20 layer specific cell types. And from these papers, what was mapped out were about 273,000 accessible chromatin peaks in this cell type specific manner from human motor cortex. So once we map that to our phenotypes, you can see our brain regions on the left here. We also threw in some controls, and all of those cell type specific regions here, non-neuronal excitatory and inhibitory neurons. And basically the asterisks show significant enrichment of these accessible chromatin types per cell type for our regions. And I don't want you to squint and think, okay, where's my favorite region? Where's my favorite cell? This is sort of just a schematic summarizing these results, where we found essentially that for the morphology of the parietal cortex was enriched for these accessible chromatin sites in astrocytes and inhibitory neurons. Our favorite light purple region here was enriched for microglia and excitatory neuron regulatory sites. And the motor cortex, which I really loved, I thought this result was really intriguing, was enriched for these accessible chromatin sites in oligodendrocyte precursor cells. And the motor cortex is actually known to have a high level of intracortical myelin, and oligodendrocytes myelinate axons. So this was a really interesting result. And just bridging one final layer here, we also found that more than a quarter of the significant loci that we found had pleiotropic effects. So they actually, these SNPs impacted more than one cortical region, which happens quite a bit in different neurodevelopmental programs. And there's two SNPs I want to highlight here that both decreased area of the motor cortex while increasing thickness of that perisylvian Broca's region. And these SNPs actually map onto MSRA, which has been shown last year to have really important implications for modern day human language. So now we're bridging the morphology of these regions that are important for both speech production and perhaps articulation with the motor cortex, and these kind of human specific regulatory regions and the MSRA gene that may have given rise to some of these unique human qualities like language. So in sum, a genetically informed atlas boosts discoverability of the genetic variants underlying cortical morphology. And we see that a lot of these, the significant loci that we find underlying cortical patterning come from the multiple neurodevelopmental programs. We have some conserved loci, but sort of the take home of this talk is that there's a select few regulatory features, some of which may be specific to modern day humans, that even though they're small tweaks in the genome, have had very large or widespread downstream effects on different regions of the brain. And perhaps these regulatory features are the key to understanding human specific traits and disorders. So with that, I'd just like to thank you. I'd like to thank the Kavli for funding this work. All of my co-authors are listed here. And I was also supported by some grants from Canada. And I welcome any questions. Thank you so much.
Thank you. Excellent. Questions from the audience?